today's lesson, we're going to be looking at factors that affect enzyme action, and specifically the rate of reaction. So for this lesson, the first part, we're going to explore how temperature and pH affect enzyme action. I'm also going to talk about substrate concentration as well. And we're going to see how various conditions regarding temperature, pH, and even the substrate concentration can affect the rate of reaction. The second part, we're going to look at how different enzymes work at different optimum temperatures and pH values. Now, this will lead on to the following lesson after this, where we will look at, again in a bit more detail, digestive enzymes and making digestion a bit more efficient. So this lesson we're just going to look at factors that affect enzyme activity. Starter questions that I want you to answer before we start with this lesson. So on Teams, I'm expecting you to give your answer. And can you pause the video here and can you write the answer in your notes, but at the same time can you also type your answer or send your answer across onto the Teams channel. So the first one is what name is given to the region where a substrate will bind. So I'm just asking in terms of the enzyme, the region where the substrate will bind, what is the name that we give to that region? And question 2, give 2 mechanisms for enzyme substrate reactions. Now, I don't want you to describe the mechanism, I just want the name of 2 mechanisms for enzyme substrate reactions. So just pause the video here and can you post your answers back on the Teams channel. We're going to talk about 3 specific factors that will affect the rate of an enzyme controlled reaction. And we need to know by the end of the lesson why pH, temperature, and substrate concentration affect enzyme activity. But first of all, we need to understand that there are certain factors that do affect enzyme action. So we are going to look at 3 of them. So can we just write down here the rate of an enzyme controlled reaction the rate of an enzyme controlled reaction can be, let's say, altered via, and we're just going to state the 3. Okay, so the rate can be altered, so it can be changed. It can either be increased, you can either increase the rate or decrease the rate, or completely stop the reaction by altering these 3 conditions that we're going to look at. So 1 is substrate concentration. Okay, 1 is substrate concentration. 2 is temperature. Okay, and let's third one. And the third one we'll say is pH. So by altering these 3 factors, we can change the rate of an enzyme controlled reaction. Now, if you do biology A level, you will have to also know about inhibitors as well. For example, there are competitive inhibitors and non-competitive inhibitors. So these are factors that if you pursue biology further, you will have to look into, we'll have to study in a bit more detail. But for now, substrate concentration, temperature and pH, these are factors that you need to know about in terms of how it may affect the rate of an enzyme controlled reaction. Now, in terms of your exam, most of the questions tend to revolve around these two here. Okay, so when we talk about temperature and pH, but it's good to know about substrate concentration because it's the underlying principle by which we can understand temperature and pH. So that's why I'm going to give you a brief explanation about substrate concentration and its effect on enzyme controlled reactions, and then it's easy to understand temperature and pH. Before we proceed to look at how temperature, pH and substrate concentration affect enzyme activity, we first need to understand what is happening at the enzyme substrate level in terms of how a reaction comes about. Now, this is due to something known as collision theory. So, the molecules the enzyme and substrate must have sufficient kinetic energy when they collide. So the first thing is that they must have sufficient kinetic energy when they collide, when collision occurs. If they don't have sufficient kinetic energy, a reaction or product cannot be formed. Now, I'm not going to state what the kinetic energy is right now. I want you to think about that as we go through the lesson. It's one of the three. Where do you think the kinetic energy is going to be important? Which one of the three? So I'm just stating here collision theory, just an overall picture of how enzyme substrate reactions occur. The first one is the enzyme and substrate must have sufficient kinetic energy when collision occurs. Okay, if there isn't sufficient energy when the substrate and the enzyme collide, no product will be formed. The substrate will not be converted into a product. Okay, the next one is that collisions must be in a specific orientation. Okay, so what do I mean by a specific orientation? So when two objects collide, and we're looking at it in a three-dimensional, let's say, space, two objects, when they collide with each other, they can collide head-on, for example. They can collide just a glancing, let's say a glancing blow or a glancing touch as they move past each other very quickly. Or they can collide, one collides with the front or one collides with the back. And these are different orientations in terms of collisions. Okay, so why is this important in terms of enzyme substrate reactions? So I've said that collisions must be in a specific orientation, i.e. they must collide in the correct manner in terms of their orientation to each other. So if I was to show you this diagram here, where we can see that two particles are colliding. One is a head-on collision, and in this case it's more energetic, and a glancing blow or less energetic collision is not going to produce a reaction. A product cannot be formed. Now, my question here is, why is this important when we talk about enzyme substrate or enzyme controlled reactions? Why is this important? Why is a specific orientation important when we have an enzyme controlled reaction? And I want you to pause the video here, and I want you to think about why the orientation of collisions are important when we talk about enzyme controlled reactions. And I want you to post the answer back on Teams. Why is it important that we consider orientation when we talk about enzyme controlled reactions? We talk about the effect of concentration on an enzyme controlled reaction. We have to talk about the nature of the collisions and how often they happen. And this is directly affected by the concentration and temperature. So what we say is, for a chemical reaction to occur, there must be successful collisions that occur frequently. So if we are to make use in terms of a biological context of chemical reactions, for example, metabolic reactions, there must be successful collisions, but they must also occur frequently. And this word frequently is important, specifically when we come to look at temperature. And it's also going to be relevant here. So I'm going to put down just a sentence here, and I want you to pause the video and think about how this is going to affect the rate of an enzyme controlled reaction. So let's say if you have low enzyme or substrate concentration. So what we've got is a low enzyme or substrate concentration. Let's say we have a beaker and we've got X amount of substrate. Let's say we've got 4x amount of substrate and we've only got 1x amount of enzyme. How is this going to affect the rate of a reaction? So just pause the video here and think about how this is going to affect the rate of a reaction. Will it be, if we have a low concentration, what is going to be the effect on the rate of a reaction based on the statement above? So what you should have is that with a low enzyme or substrate concentration, this will result in a slower chemical reaction. Will result in a slower chemical reaction, i.e. the rate will be slower. Now, this should be fairly obvious. It should be quite easy to grasp from our explanation here. But if you're struggling to visualize what's happening, I'm going to show you a simulation. And this simulation deals with gas particles, but you can use it to model how enzyme substrate reactions also work. So let's take a look at this simulation. So in this simulation, as you can see, this is going to be more for gas particles. However, we're going to simulate how enzyme substrate reactions would occur in terms of the effect of concentration. So our purple particle represented here, you can see the two particles represented near the bottom of the screen. So the purple particle is going to be our enzyme, and our red particle is going to be our substrate. So let's just put in our enzyme. So we'll just put in one enzyme for now. So let's see if we can put one in. Okay, there's this many enzymes. We'll just keep it at this. All right. So we've got these enzymes in our container. And as you can see, they have a certain amount of kinetic energy and they're moving around. Now, if a reaction is to occur, actually, let's start this again, because let's put in one. Here is one enzyme. Okay, one enzyme has gone into our container. Now we have to have a quick rate of reaction. Our goal is to obtain a quick rate of reaction. So if we put in a large amount of substrate, so we're trying to get a large amount of let's say a quicker rate of reaction, though we have the substrate colliding with the enzyme, and we can see it happening. All right, so as the particles collide, let's say as it collides with the enzyme, a chemical reaction is taking place. So let's say each collision is occurring in the correct orientation, even though that wouldn't be factually correct. But let's say, for argument's sake, each collision with that enzyme produces a product. But because of the low concentration of the enzyme, you can see that it doesn't happen that often, or not as often as we would like, specifically for biological enzymes or metabolic pathways where you need a quick rate of reaction, because there's not enough of the enzyme substrate collisions happening at any given time. Okay, now what happens when we increase the enzyme concentration? Look at how many collisions are occurring when we increase the enzyme concentration. So we've kept the temperature the same, but here you can see very clearly that the number of collisions occurring between the enzyme and the substrate, there are more of them. The frequency of the collisions is more. Therefore, we're getting a quicker rate of reaction. So, and it's the same thing for substrate. So, let's say we were to have a lower substrate, and it's not easy to model the substrate reaction, the effect of substrate concentration, simply because when a substrate reacts with an enzyme, if a substrate was to react with an enzyme, it will produce a product, and the product is now different from the original substrate. So to model it here would be slightly incorrect, in the sense that you would not have the substrate remaining unchanged. If you remember back to our first definition, enzymes remain unchanged. That's why we were able to model that single enzyme going around. And it's not the best model to use to show enzyme substrate collision. However, you can see the effect of concentration, i.e. that the lower the concentration, there are less successful collisions happening at any given time. So here you can see a graph that's representing substrate concentration and initial rate of reaction. What I want you to do is, based on our discussion so far and the modeling that we've just looked at, I want you to construct an answer that describes what is happening in terms of substrate concentration for this reaction that is controlled by an enzyme. This is an enzyme catalyzed reaction, and I want you to describe what this graph is showing. So explain this graph or describe the pattern that you're seeing on this graph. And I want you to use certain keywords. I want you to use the word initial in your answer. Active site. What else can we have here? Enzyme. You need substrate. What else can we have? Increase. Okay, so we'll leave it here. You can use other keywords, but I want you to within your answer, these words must be there. So I want you to construct an answer to describe this graph in terms of how the concentration, substrate concentration, is affecting the initial rate of reaction. In the previous simulation, we were looking at enzyme effect. So it's slightly different. But here, I want you to now use the same principle that we were looking at. Now apply it to substrate concentration, and this graph should help you. Okay, so I want you to construct an answer. And so pause the video here and construct an answer for this graph that's in front of you here, that's displayed here. Okay, and using the keywords that are listed to the side. Look at an answer. So what we say is that, first one, I'm going to bullet point it. I'm going to try and condense it down. So by looking at this answer, you can expand on your notes or the answer that you have written down. So as substrate concentration as substrate concentration increases, okay, the initial rate of the reaction also increases. So as a substrate concentration increases, the initial rate of the reaction also increases. Now you can see that here. Let's turn the ruler around. Okay, you can see that here. So where there's an increase, in the rate, so you can see that as you increase the substrate concentration, the rate of the reaction is also increasing. Now, the next point, if there are more substrate molecules around, then there's more chance that the enzyme's active site can bind with a substrate, can successfully bind. So this allows for more successful collision and binding of substrate with enzyme active site. Okay, so just remember that we need a successful collision in terms of orientation. This is what we were looking at previously in the lesson, where we were talking about why orientation was important, because the active site is a specific region on the enzyme. Remember back to our starter question about, I asked you to remember the specific region on an enzyme that is responsible for a chemical reaction. It is the active site. Therefore, the active site and the enzyme need to collide in a specific orientation and successfully for the reaction to occur. And if you want this to happen at a fast rate, this binding of the enzyme and the substrate needs to happen frequently. And if it needs to happen frequently, you need to have more of the substrate around. By increasing the substrate concentration, you increase the chance that the enzyme and the enzyme's active site will bind to a substrate. All right, so that's what is being represented on the graph there. As we increase the concentration, the initial rate of the reaction also increases. However, if you keep adding more substrate concentration, and then a point will come where all the enzyme's active sites are occupied and continuously working. This means that when you add more and more concentration when you keep increasing the concentration of the substrate, you do not get an increase in the initial rate. And that is represented also on the graph. So you should notice here that we start to get a leveling off. So, and also here at this point, you can see, okay. You can see at this point, there is a leveling off, a plateauing of the graph. Now, at this point, the velocity of the reaction or the initial rate of the reaction cannot be increased. This is because all of the enzyme's active sites are occupied and the enzyme is continuously working at any given point. So at any given point in time, once you've reached a certain amount of substrate concentration, you are occupying all the active sites on the enzymes, and the enzymes are continuously working. Therefore, at this point, adding more substrate will not increase the rate of the reaction. And this point is known as Vmax. And you can see it here, Vmax. Vmax is the velocity. V stands for the velocity of the reaction, how fast the reaction is proceeding. And at Vmax, you can see that adding more substrate will not increase the rate of the reaction. So let's put this down in a concise way. So let's just say adding more substrate will increase the rate up to a certain point. Okay, so adding more substrate will increase the rate up to a certain point. Okay, and this is Vmax. Vmax is the point at which all the enzymes are continuously working, i.e. all the enzymes' active sites at any given time are occupied. And therefore you're going to have a backlog of, or let's say a queue of substrates waiting for an active site to be available. And that's why adding more substrate concentration does not increase the initial rate. So when we talk about the effect of concentration on enzyme controlled reactions, and specifically substrate concentration on enzyme controlled reactions, you will see this shape of graph. Now you will notice that when we come on to look at temperature, the graph is different. So if I was to ask you to compare two reactions happening for an enzyme and two factors that are being altered, and I would ask you to identify just based on the graph, you should be able to do that. So take a note of the graph here. look at the shape of the graph, look at the plateauing occurring, and look at that if you continuously increase the substrate concentration or this particular factor of substrate concentration, look at what's happening to Vmax or the initial rate. Just a final point on the graph, the regions where the two lines have been indicated, here and here, and also this region here, you can use specific words to describe the pattern of the graph here. You can use a word that there is a directly proportional relationship at these 2 points, where you can see that as you increase the substrate concentration, how the initial rate of reaction is increasing. But then you can say that as the substrate concentration is continuously increased, we start to get a leveling off and then a plateau being reached. And why this is important, because what's happening is that the substrate concentration, as I mentioned earlier, is finding it difficult to find a free active site, and that's why you see this leveling off, and then you should use the word plateauing at Vmax. So there is a plateauing where the substrate concentration may increase. So on the x-axis we can see that the substrate concentration will continuously increase, but the initial rate will not, because the graph has plateaued, and this is because all the active sites are occupied. So just be aware that you can use these words when describing a graph of this nature. At how substrate concentration affects enzyme activity. Now we're going to look at the effect of temperature. And again, we have to think about the enzyme and the substrate in terms of collisions. So we've already looked at the aspect of the enzyme and the substrate colliding enough times for a rate of reaction to be sufficient for biological systems or metabolic pathways. So the number or the frequency of the collisions is important, and we can increase that by increasing the number of substrate molecules available. Another way in which we can increase the frequency of collisions that take place is by increasing the temperature. So the effect of temperature on enzyme activity, one would be it increases the frequency of collisions between substrate and enzyme. Okay, so it increases the frequency of collisions between substrate and enzyme. Now, so we are getting more collisions happening because the molecules have greater kinetic energy. Greater kinetic energy is because of the increase in temperature. We supply thermal energy to a system, and the particles or the molecules in that system gain that energy, and we see kinetic energy occurring. So we see that kinetic energy, and that translates to more collisions. So we say that the effect of temperature is to increase the frequency of collisions between substrate and enzyme due to greater energy, and we will say it's kinetic here, of molecules. And how did the molecules get this kinetic energy? It would have been due to the higher temperature. So we say that the effect of temperature is ultimately increasing the frequency of the collisions between the substrate and the enzyme due to greater energy that the molecules have. So we looked at the simulation previously and you could see that the molecules were moving. So this movement energy is the kinetic energy, and by raising the temperature we are raising the kinetic energy, the movement of the enzyme and the substrate, and this means that overall, on average, more collisions are occurring between the enzyme and the substrate. The second point, by increasing the temperature, we allow the enzyme and substrate to collide with let's say greater energy. Now this is slightly different from the first point. In the first point we're saying that we're increasing the frequency of the collision, so the collisions are occurring more collisions are occurring at a given time. The second point is that the particles now are also colliding with greater energy. This means that it makes it more possible and more probable that bonds will be formed between the active site and the substrate. And this makes it more probable that a product will be formed because the collisions are of a certain amount of energy. So you need a certain amount of energy for the collision between the enzyme active site and the substrate to form a product, for the bonds to form. So by increasing the temperature, we allow the enzyme and the substrate to collide with greater energy, thus allowing for bonds to form or break. So depending on the type of reaction we are looking at. So by increasing the temperature, my apologies, at the top I should say the effect of an increase. If we look back here at the title, the effect of increase. Okay, so if we're increasing the temperature, this allows for the enzyme and the substrate to collide with greater energy, thus allowing for bonds to form or break. And this means a chemical reaction can take place. So I'll just model this again on that simulation. We can see what the effect of temperature is, and I'm sure by now you have a good idea of what will happen in terms of the collisions between enzymes and substrate. Here you can see the simulation showing the enzyme and the substrate. So just a reminder, the purple spheres are the enzymes and the red spheres are the substrates. Now, what you can see is that the temperature is quite low. It's at 62 Kelvin. Now, you can see here, so if you study this simulation and see how many times the collisions take place between the enzyme and the substrate. Now, what you can see is that for a low temperature, and bear in mind we're saying that every time a collision occurs, it is successful. This is not always the case, but for this simulation, for argument's sake, we will say that every time a collision occurs, there's a successful collision. So just take a look now for a moment and see how often, let's say for 5 to 10 seconds, how often a collision occurs at 62 Kelvin. Okay, now I'm going to increase the temperature. So you can see the temperature is rising. And look at the motion of the molecules now. Now notice how often a successful collision is occurring. Notice we are keeping the substrate concentration the same and the enzyme concentration the same. So if we increase the temperature a bit more, and you can see that the frequency of collisions is much greater. We can go a bit further. So what we are essentially seeing is that as the temperature increases, the molecules, the substrate and the enzyme have more kinetic energy. And this makes their collisions happen at a higher frequency and more chance that successful collisions will occur, i.e. there's more chance that the substrate is hitting the active site simply because more collisions are taking place. But at the same time, the particles, if we look carefully here, we can see that they're moving much faster. This means that the collisions are occurring with greater energy. And this means that bonds can be formed and bonds can be broken, i.e. a reaction can take place. So let us now just sum up what happens as the temperature increases and we keep increasing the temperature. We have seen how it affects the kinetic energy of enzymes and substrates. Now we're going to just expand this further. So the first point is that the rate we'll say that the rate of enzyme substrate reactions or enzyme substrate collisions, let's say, increases as the temperature increases. Now, I want to add one word here that I've just missed out. I just noticed that we've said that the rate of enzyme substrate collisions increases as the temperature increases. So we're just stating what we've just observed. But could you write here that the rate of successful enzyme substrate collisions increase as the temperature increases, because what we're going to see later on is that collisions may be taking place but no forward reaction is actually happening, and we'll see why in a moment. So as we increase the temperature, the rate of successful collisions also increase. Now once we get to a certain point, and for most biological systems this is around 40 degrees. Now for the human body it's approximately 37 degrees, at which the enzyme and the substrate will be colliding successfully and they will be working at their most optimum rate. Once you go past, let's say 40 degrees, you will start to see a decrease in the rate of reaction. Now this is different from increasing the substrate concentration, because if you remember, the substrate concentration, if you kept increasing it, it didn't decrease the rate plateaued. However, when we talk about temperature, when we keep increasing the temperature, and in this case let's say 40 degrees and above, we will start to see the rate of reaction for an enzyme controlled reaction. We'll see that rate gradually start to fall, and it will fall, if you keep increasing the temperature, it will fall to 0. So this is the difference between the effect of temperature on enzymes. This is because after a certain temperature, and this is different for each type of enzyme, the enzyme begins to break down, and the word we use is denatured. The enzyme becomes denatured. This means that the tertiary structure, the 3D folded proteins or amino acids, their bonds have broken down. The hydrogen bonds or other bonds that hold the protein tertiary structure together have broken down. And this is what we mean by denaturing. Do not say that the enzyme has been killed, or do not say the enzyme has died, because the enzymes are just molecules that have a specific shape and a specific structure, and by increasing the temperature we break apart that specific structure and the specific bonds that hold the tertiary structure together, and by breaking that apart the enzyme will stop working. Now what I want you to do is I just want you to pause the video here, and I want you to write down what specific aspect of the enzyme would have been damaged that causes no more reactions to take place. So listen again to what I'm saying. I'm saying that we've increased the temperature and the enzyme has been damaged. It's been denatured, its tertiary structure has been collapsed, and bonds that hold the 3D structure together of this enzyme molecule have been broken down. Now, what specific aspect of the enzyme would have been affected by the denaturing process that caused the rate of reaction to drop to 0? So can you just write down a sentence explaining that? Okay, what you should have actually put down is that the active site on the enzyme would have changed its shape or lost its shape. And this means no more substrate molecules can bind successfully to the enzyme. And so because the enzyme and substrate cannot sufficiently bind because the active site has lost its shape, because the bonds holding the 3D structure have collapsed, the enzyme has said to have been denatured and stops working. So let's put this down into words. Most biological enzymes have an optimum temperature which is approximately 37 degrees. So we say most biological enzymes have an optimum temperature of 37 degrees, of approximately 37 degrees. Okay, and for humans it is 37 degrees. Now, they have an upper limit of 40 degrees. So then we say that the upper limit for most biological enzymes is approximately 40 degrees. Now you may notice that I'm using the word approximate. This is because there are many types of biological enzymes and not all of them have specific optimum temperatures. They can vary, and this is the case also for pH, that there are no specific set pH values for all enzymes. Enzymes based on their function, based on their structure, will work better at various pHs. So what's optimum for one enzyme may not be optimum for another. But when we talk about biological enzymes, yes, there may be certain temperatures where they work best at, but there is essentially a limit, almost like an upper limit, where once you pass that limit, biological enzymes are not going to function. This is because, as we've just explained, that the protein structure that holds the active site together starts to collapse, starts to denature. So the upper limit is 40 degrees. Passing, let's put it another way. Okay, let's put it this way. Passing this temperature, so going higher or above this temperature, the enzyme begins to denature. Okay, let me spell that a bit better so you can see. Starts to denature. So what we're saying is that after approximately 40 degrees, the enzyme is the rate at which it's going to be able to work is going to be reduced, and eventually the enzyme will stop working altogether. Now, why is this happening? So now we know what's happening in terms of after a specific temperature, what's happening to the enzyme, but why is this happening? So let's look at why it's happening. Now, the amino acid chains, so the long amino acid chains that hold the enzyme together begin to unravel. Okay, and this means that the shape of the active site changes. So if an enzyme is denatured, the long amino acid chain, the long amino acid chains begin to unwind or unravel. Okay, unwind, or let's say unravel. Notice here we can see an enzyme and its highly folded three-dimensional shape and how it's held together via various types of bonds. Now, you can see the coil structure, and we came across this last lesson where we were looking at an enzyme in a three-dimensional space. I want you to notice how compact the enzyme is and how it's folded together and how many coils there are. So when we denature an enzyme, I'm going to show you an example of a denatured enzyme, and I want you just to compare what you're seeing here to an example of a denatured enzyme. So here you can see, notice that the various foldings and how tightly compact and structured this protein is. This is a type of enzyme. It's a hydrolase enzyme. Now, I'm going to show you a type of hydrolase enzyme which has been denatured. So, for your reference, so you can able to see so you can understand this process of denaturing, I want you to be able to compare an enzyme in its normal state to its denatured state. Now, this is an enzyme, another type of hydrolase enzyme that has been denatured, and it's showing this after 10 hours. Now, what you can straight away notice is that it's more loosely packed, it's more unwound and unraveled, and there are less coils available. Now, all of this affects the actual 3D structure of the enzyme, and it significantly affects the active site as well. And you are not going to get specific binding occurring because the enzyme has lost its structure. Its tertiary structure has collapsed. You can see more open spaces. Now this is because of the denaturing process. As we increase the temperature, the long chains of amino acids are collapsed and broken and begin to unravel. And this causes the 3D structure, the tertiary structure, to collapse also. And this is what causes the enzyme to stop working, because the active site has lost its specific shape and therefore it cannot bind to the substrate successfully. So here you can see two diagrams representing the denaturing process of enzymes. And you can see here in a two-dimensional way where it's simply represented that the bonds holding the coil structures together unravel, and you can see the open spaces. Let's put it in red. You can see there are more open spaces, and this is because the coil structure has also collapsed. You see these coil structures have collapsed and there are more open spaces, and this is what results. The bonds holding, there are specific bonds, hydrogen bonds and other types of polar bonds that hold this structure together, and when they collapse, the protein begins to unravel and unfold, and temperatures attack these bonds and break these bonds, and as a result you get an unfolding occurring. Now, so going back to our sentence, let's finish this off. So as we said, if an enzyme is okay, so I've just noticed here, with that sentence, we've said if an enzyme is denatured. Now, the enzyme is undergoing a process of denaturing. So it's denatured once it's fully unfolded. So just a slight note there. Just note that it should be that if an enzyme is denaturing, the long amino acid chains begin to unwind or unravel. Because once it's denatured, just be aware that's when it's already unfolded. So this should be denaturing. So let's amend. Okay, just in your notes, just correct it slightly, that it should be if an enzyme is denaturing or if an enzyme is breaking down, the long amino acid chains begin to unravel. And what we'll say is that this results in the active site losing its specific shape. Therefore, the substrate will no longer fit. Okay, so as the protein is unfolded, because the long chains of amino acids are unwinding, unraveling, the active site loses its specific shape, therefore the substrate will no longer fit, and therefore a reaction cannot proceed. Here we're looking at two diagrams where the effective temperature is being represented. For your notes, I want you to take down one of the two diagrams and make sure that you annotate your graph clearly. And what I want you to notice is that on both graphs, that it does not, in terms of reaction rate, come down to 0. Okay, so the graph does not represent a reaction rate of 0 at 0 degrees. Now this is important. On substrate concentration, you clearly saw when there is no substrate concentration, there can be no reaction. However, here, when the temperature is at 0 degrees, we are still saying that a reaction could technically occur. Now, this is because at 0 degrees, the particles or the molecules still have some form of kinetic energy. If we go back to the simulation, we were saying that particles or molecules must collide to have a reaction take place. So there must be some kinetic energy, movement energy there. And we have not, as of yet, got down to absolute zero. And at absolute zero, it is said that at that temperature, the molecules or particles will stop moving. It's hypothesized. Now, that's why here you don't get the rate of reaction dropping to zero at 0 degrees, simply because one, the particles or the molecules are still moving, and there could very well be a reaction, or it will be very the rate of reaction will be very low, and it will be much slower, but there potentially could be. Furthermore, there are certain bacteria that work effectively at 0 degrees and sometimes lower than 0 degrees. So that is why we don't say that at 0 degrees the rate of reaction is 0, because when we talk about temperature and its effect on collisions between substrate and enzymes, we're essentially talking about the movement of enzyme and substrate, and you have to lower the temperature to absolute zero, and that's not 0 degrees Celsius, it's further down. It's much further down. It will be somewhere much further away. So this is a bit more than what you need to know at GCSE biology, but you just have to just appreciate that the graph does not go to zero at reaction rate. The reaction rate does not go to zero at 0 degrees. So first point is because molecules are still moving at 0 degrees, there is still some movement, and also some living organisms are able to work effectively at 0 degrees. So as I said, enzymes are not fixed at one specific temperature where they have an optimum range. They have a wide range based on the type of enzyme we're dealing with. So what I want you to do is take down one of the two graphs, make sure you clearly annotate it, and you can clearly see that, or you can clearly understand that after a certain temperature, you see here, the actual enzyme stops working. It is denatured, and you should be able to explain this type of graph without any labels present. So pause the graph, study the graph. So pause the video, study the graph, and copy it down in your notes, and make sure you're able to explain the graph that you're seeing without any annotations to help you. Shape of a protein molecule is essentially held together by forces that are exerting themselves or interacting between different parts of the protein molecule. Now, because the protein molecule, as you've seen in the 3D simulation, it's folding on itself and in between and coming into contact with other protein strands. So what happens is the shape of the active site of any particular enzyme is a result of the forces between the different parts of the protein molecule. So can we just write this sentence down first? The active site shape of an enzyme. The active site shape of an enzyme is due to different forces. Let's put various. is due to various forces between different parts of the protein molecule. So it is these forces between the different parts of the protein molecule that give rise to the specific shape. And as you were looking at the simulation, you were seeing how long protein amino acid chains were coiled, folding on themselves and coming into closer contact with other longer coiled protein chains. And what happens is that this creates, because of the nature of the atoms that make up protein molecules, this creates forces of attraction between the protein chains. Now, in chemistry, you may have come across from previous lessons, you may remember where I was talking about how not all bonds are equal, are held equally. For example, an oxygen atom will hold on to the bonds in a covalent bond much more strongly than a hydrogen atom will. Now, this causes a certain level of polarity to start to occur in bonding, and this can cause certain regions of, let's say, charge to be different. And this causes, and when we put this onto a whole molecular scale, and where large protein molecules, large protein chains are folding on themselves, we can get different interactions between the various atoms, because proteins are essentially made up of nitrogen, and they're also made up of carbon and sometimes oxygen. So we have these different atoms that hold on to electrons in varying degrees and in a bond, and this can create a certain level of electronegativity, so to speak, and this is what gives rise to the specific active site shape. Now, pH and temperature break apart these bonds. Now, the effect of pH is very similar to the effect of temperature, in the sense that the active site shape is slowly broken down, is slowly collapsed, and therefore the enzymes will stop working. However, what you will find is that with pH, the range at which the enzyme will work is much narrower. And this is what you're going to see shortly when I show you the graph. But let's finish off our explanation first. So the active site shape of an enzyme is due to the various forces between different parts of the protein molecule. And these forces hold the folded chains in place. So can we just write these forces hold the amino acid chains, in place. Okay. Now, pH breaks these forces. So what we'll say is that, it shouldn't be pH breaks, it should be drastic changes in pH will break these forces. So can we write drastic changes in pH for a particular enzyme will break these forces. So what is being said here is that the reason I've said for a particular enzyme is that enzymes, when we come to pH, show a larger range in terms of their ability to work. So let's say enzyme A can work at pH 2 to 3, whereas enzyme B may work at pH 7 and 6 more effectively. So that's quite a large range. However, when we talk about the enzymes in general, they are quite narrow in terms of their tolerance level. So an enzyme that's designed to work at pH 1 and 2, you will find does not have a large tolerance level when it comes to pH. Whereas in temperature, what we were looking at, we could see that the curve was quite spread out. Now when we come to look at pH, we'll see that it's a bit more narrower, indicating that enzymes are highly sensitive to pH ranges more so than temperature. So drastic changes in pH for a particular enzyme will break these forces. This results in their active site shape being lost. And you can finish off this sentence explaining if an active site shape is lost, what happens to the enzyme substrate complexes that should be formed. What can happen if you've lost your active site shape? What does that mean for the substrate binding? So you can finish off that sentence, just maybe a sentence or two explaining what that means, because I've explained it twice now. Now I want you to finish off that in your note. So I've talked about what's happening at pH. It's essentially the same way losing the active site shape, and this results in the enzyme not being able to bind onto a substrate successfully. So now I want you to take down this graph for the rate of reaction when pH is a factor. And what I want you to notice is the region of the graph where pH is 3 and pH is approximately coming close to 11. Notice that the rate of reaction has dropped to 0. Now, why this is important is because we can compare the pH graph with the temperature graph. So if you were asked to identify which was a pH controlled enzyme reaction graph or a temperature controlled enzyme reaction graph, you should be able to make that comparison, because if you remember, for pH, putting these graphs side by side, temperature and pH, you can see that the differences in these graphs. You can see that there is a narrower range for pH as opposed to temperature. And if you are asked to identify these graphs, just based on the based on the shape of the curve, you should be able to do that. And if you weren't given any labels for the graph. Now, what I want you to, looking at the graph for pH, I want you to give me an enzyme, the name of an enzyme that works best at pH 3. So for pH 3, okay, can you give me the name of an enzyme? So name an enzyme that works best at pH 3 within the digestive system. So part of the digestive system, which enzyme would work well at pH 3? And let's give it a range. Let's give it a 1 to 3. Let's just make it 1 to 3. Let's say 1 to 3. Let's make it 1 to 3. And for pH 7, what enzyme would work best at or work around pH 7? So could you name the enzyme here and name the enzyme there? Part of the digestive system. And could you type your answer back on Teams?